Perfect. Dr. Whelan, or are you going to introduce him?
Yes.
Perfect.
So just let me know when you're set.
Yeah, we're ready to go.
Okay. Dr. Whelan, you ready?
Yes.
All right. Well, good morning. Welcome to Medicine Grand Rounds via Zoom. Today we continue our lecture series on COVID-19. And as a reminder, please use the chat function on Zoom to ask questions, and we'll have a Q&A after the talk that I'll lead. I'm excited to announce that today we are joined by the current head of the Department of Molecular Microbiology and the Marvin A. Brennike Distinguished Professor of Microbiology, Dr. Sean Whelan. Originally from Leeds, he received his degree in biological sciences from the University of Birmingham before joining Dr. Jeffrey Ullman's group at the University of Reading for his graduate studies. He then journeyed to the University of Alabama at Birmingham for his postdoctoral studies on the vesicular stomatitis virus. Most recently, he was a professor of microbiology and immunobiology and the head of the virology department at the Harvard Medical School. Perhaps this is a candidate for understatement of the year, but he has run a very productive and successful lab that focuses on the biology of negative strand RNA viruses. By using VSV as a model system, his lab has made numerous contributions to the understanding of viral gene expression and virus-host cell interactions. Most notably, his group was able to identify the cholesterol transporter NPC1 as an entry site into human cells for the Ebola virus. He has also worked to define a pathway by which the rabies virus is able to enter a neuron. For his work, Dr. Whelan has been well recognized. He's a 2005 Burroughs Wellcome Fund investigator, a 2013 fellow in the American Academy of Microbiology, and a 2015 NIH Merit Award recipient. He has been and remains an advocate for diversity and community engagement in science, and it is my distinct pleasure to turn it over to Dr. Whelan as he presents SARS-CoV-2, Virology and Countermeasures.
Thank you. So what I'd like to do this morning is give a little bit of a hybrid summary of what we know about SARS-CoV-2 and some additional summary of some work that has been going on here at WashU, including work from my own group. So I should start out with the following disclosure. I don't have any current conflicts of financial interest, but I have reported an invention disclosure on one of the viruses I will be talking about today, VSV SARS-CoV-2, as a BSL-2 reporter of SARS-CoV-2 infection. And I'm also an investigator on a pending cooperative research agreement with Vir Biotechnology that may have a relationship or build on some of the work that I'm talking about today. So I've been very interested in emerging and re-emerging infections of humans for many years. And this is a slide that I adapted from Tony Fauci, who uses this, a variant of this slide to emphasize some of the current threats that are present to human health from emerging and re-emerging infectious diseases. And I've modified it in a couple of different ways. One is I've added this virus, which originally was called Wuhan virus, but is now known as SARS-CoV-2. And I've also highlighted by these asterisks all of these emerging infectious or re-emerging infectious agents that are caused by enveloped RNA viruses. So one of the challenges that we face in studying emerging viral infections of humans is when a new disease emerges, one needs to be protective of oneself when you're working with the disease. And in fact, this is, or the disease-causing agent, this is my friend and collaborator, John Dye, working with Ebola virus at the US Army Medical Research Institute of Infectious Disease at Fort Detrick. And in a BSL-4 lab. And thankfully, SARS-CoV-2 is not BSL-4, but it is BSL-3, which requires a respirator to be able to work with it, as well as a suit, a Tyvek suit, and one works with it in a standard biosafety cabinet. But there are many challenges in working with infectious disease, emerging infectious diseases. One is that there's been a new emerging infection of humans every 6 months since 1980. A second has been that it's been difficult to predict barriers to cross-species transmission. There are, as I just mentioned, biosafety challenges to studying these pathogens. And as we all know, people are social, and therefore infectious diseases readily get disseminated amongst the human population. And there's also a challenge with the incubation period of an infection, the period over which people are asymptomatic but can still spread the disease. It is a concern currently for SARS-CoV-2, as I'm sure you're all aware. So a number of years ago, the World Health Organization put together a list of priority diseases that it considered to be a potential major threat to human health. And again, these are all enveloped RNA viruses. They include Ebola, Marburg, Lassa, and MERS and SARS. But notably, what the World Health Organization was advocating for is that a research and development blueprint should be focused on these infectious agents, but also disease X, which represents the knowledge that a serious international epidemic could be caused by a pathogen currently unknown to cause human disease. And disease X, as I don't need to emphasize, is now SARS-CoV-2. So this is a slide that I took from an article, an interesting article from the Gates Foundation about the role of research and development during a pandemic. And I think it really quite effectively summarizes what people have been working on since we became aware of this virus at the end of last year. And so there's a great deal of effort going on to improve testing. We need efficient testing of a large number of people in the population in order to know where the virus is and who's been infected, who's recovered from infection by using serological testing, and who may be protected against reinfection. We need to develop therapies that can combat the infectious agent. So treatments that include both small molecule inhibitors, drugs, antibodies, and other potential therapeutic agents. And we need to ultimately prevent the infection through the use of vaccines or through therapeutic antibodies that could be given prophylactically. So I'm sure many of you have seen the Johns Hopkins University website that summarizes the global cases of coronavirus. And as of last night, there were 3.7 million total confirmed cases, with 263,000 deaths reported around the world. And here in the United States, we can see that the virus is essentially everywhere, although there are hotspots in the population dense parts of the country. And there are also additional hotspots here in the southern part of Georgia and in Louisiana. And there's a little bit of a darker spot here in St. Louis that represents an increase in people who've been infected by the virus. So what are the symptoms of the infection? So some of the common symptoms are fever, dry cough, fatigue, but also a loss of sense of taste and smell. There are a number of uncommon symptoms: headache, nasal congestion, sore throat, coughing up sputum, shortness of breath, pain in muscles or joints, chills, nausea and vomiting and diarrhea. And in severe disease, One can have high fever. There can be coughing of up of blood. You get decreased white blood cells, and you can also have kidney failure. So where did this virus come from? So if we look at what our current model of coronavirus evolution is, I took this from an article from Patrick Wu and colleagues published in Journal of Virology in 2012. But SARS-CoV-2 is in a class of coronaviruses called the beta coronaviruses. And it's thought that these viruses have a common ancestor about 300 years ago or so that was circulating in bats or birds, and that this virus basically has undergone extensive replication in bats and then has been transmitted to humans, probably via an intermediate host. The intermediate host in the case of SARS-CoV-2 is not clear. Lots of things have been speculated, but we know that that certainly seems that there has been an intermediate host in the transmission of the virus to the human population. So if we look just at SARS and MERS, these were viruses that seem to have their origin from bat coronaviruses. In the case of SARS, the virus then went through the intermediate host of civets and small carnivores, and then was transmitted to humans. In the case of MERS, camels seem to be the intermediate host for transmission to humans. And what's been seen in both cases is animal workers and healthcare workers who take care of sick animal workers or who come into direct contact with these intermediate hosts have been the folks at the highest risk of transmission. And those at the highest risk of severe disease are those with comorbidities and often that are over the age of 65. So if we look at what happened in the case of SARS-1, the virus is thought to have originated in a wet market in Guangdong province in China that involved the transmission of the virus through a palm civet to animal handlers. A healthcare worker who took care of one of those sick animal handlers ended up going to Hong Kong and staying in a hotel in Hong Kong and managed to transmit virus to many of the other guests at the hotel, who then promptly transmitted virus when they returned to their home countries, Vietnam, Singapore, and a person from Singapore then transmitted the virus to Germany. The virus was transmitted to folks in the United States, Ireland, and Canada, all from this individual that stayed in a hotel in Hong Kong. And so it just sort of exemplifies how rapidly these infectious agents can be transmitted around the globe. If we look at MERS, MERS has been detected in camels, as shown by these red asterisks, in camels and humans, as shown by these black asterisks, but then only in humans in countries highlighted by these green asterisks. And so we can again see that this virus, MERS, has become quite broadly transmitted around the planet, but not nearly as broadly as SARS-CoV-2. So what are coronaviruses? So coronaviruses are known that name, crown, comes from this characteristic appearance by electron microscopy that shows these large spikes that decorate the outer surface of the virus. These images are actually of mouse hepatitis virus, a well-studied variant of coronaviruses, well-studied in the lab. And this electron micrograph really shows beautifully these spikes that decorate the virus particle. The spike is not the only membrane protein of the virus. There's also additional membrane proteins. This membrane protein here, another membrane protein that is probably an ion channel, and then within the virion is the viral genomic RNA coded by a viral nuclear capsid protein. So how do these viruses replicate? So the virus binds to a cell surface receptor. In the case of SARS and SARS-CoV-2, that receptor is ACE2. Binding at the plasma membrane can lead to membrane fusion and release of the contents of the virus into the cell, but it depends on the presence of a protease that can also cleave the spike protein and facilitate the process of membrane fusion. We know that that protease can be TMPRSS2, a serine protease, or a closely related serine protease, TMPRSS4. If cleavage of the spike doesn't occur at the plasma membrane, the virus is internalized through endocytosis. And then endosomal cathepsins can also be involved in processing the spike and can lead to membrane fusion from an endosomal compartment. Irrespective of which of those two entry routes are used, the net result is to deliver the viral payload into the cell, which then can be used for translation of the viral RNA. The first proteins that get translated are those that encode the viral replication machinery, and they set up these double membrane vesicles within which the viral replication machinery is inserted into them, upon which the viral genomic RNA is transcribed and replicated. Transcription of that genomic RNA leads to the production of subgenomic RNAs, some of which encode the structural proteins of the virus, the spike, the envelope, the membrane protein, and the nuclear capsid protein. The membrane-associated proteins are co-translationally inserted into the endoplasmic reticulum. The nuclear capsid protein can then coat the genomic and RNA that's produced via copying of the input genomic RNA into an antigenomic intermediate RNA that then serves as template for production of more genomic RNA. That genomic RNA can be coded by the nuclear capsid protein, and the nuclear capsid protein RNA complex associates with the endoplasmic reticulum inserted membrane proteins and buds out through the ER-Golgi intermediate compartment and releases virions from the cell that can go on and infect new cells. The virus replication cycle takes place entirely within the cytoplasm of the cell, although the virus does manipulate the nucleus. So, as you all know, antibodies are one of our natural defenses against viral infection, and there are many mechanisms by which antibodies can block viral infection. So this schematic is actually of a non-enveloped virus that is attaching to a receptor on the cell surface. It then gets endocytosed and releases the contents into the cell. Or in the case of some viruses, you can get released directly from the plasma membrane. But antibodies can block this process of infection through a number of different ways. They can aggregate virus particles, which suppresses their ability to infect cells. They can also prevent engagement by the virus receptor. They can stabilize or destabilize the capsid, both of which can be negative for viral infection. They can block the process of endocytosis, or they can block the process of membrane fusion. And there are, for many different viruses, examples of antibodies that work through each of these different strategies. If we look at antiviral drugs, there are currently about 90 drugs that have been approved for use in humans against a variety of different viruses. The overwhelming majority are against HIV, but we have several for hepatitis C virus, herpes simplex virus, influenza, hepatitis B, other herpes-related viruses, respiratory syncytial virus, and papilloma virus. But if we look at how those antiviral drugs work, in contrast to antibodies that work by blocking stages, largely by blocking stages in the entry process of virus infection, only 6 of these antiviral drugs work by blocking viral entry. And of those 6, 4 of them are actually antibodies. One is a peptide that blocks the refolding stage of the HIV envelope protein, and one is a small molecule that blocks engagement of the co-receptor used by HIV to inhibit infection. The other drugs all work by targeting aspects of the intracellular stages of viral replication. And so this is quite a contrast to how our natural defense mechanism against viral infection, or one of our natural defense mechanisms against viral infection, neutralizing antibodies, works. So there are many possible interventions that are being studied to block SARS-CoV-2 infection. And some of them are highlighted on this slide. So monoclonal antibodies are certainly an area of active research here at WashU and also elsewhere that can work to block infection by targeting presumably stages in entry. There are some antibodies that block through blocking ACE2 engagement and some that work through different mechanisms. The groups of Ali El-Abedi, David Fremont here at WashU, and Mike Diamond have all been involved in studying these monoclonal antibodies or different monoclonal antibodies and how they can block infection. Also, Gaia Amarasinghe in the pathology department has been engaged in studies using monoclonals that can block infection. You must all have heard of the convalescent plasma trials that are happening here at WashU and around the country using plasma from patients who've recovered from infection that also hopefully block by inhibiting viral entry. There's an inhibitor of this protease, TMPRSS2, camostat mesylate, that is being investigated as a potential inhibitor for virus entry. And there's also, there was a lot of interest in using chloroquine and hydroxychloroquine, which work by neutralizing the pH of the endosomal compartment that the virus may be, that the virus uses for internalization. But chloroquine and hydroxychloroquine have not shown promise in clinical trials. And I think a lot of the efforts focused on these, the data is not very compelling in terms of the success in clinical trials. There's been a lot of excitement about remdesivir, which is a polymerase inhibitor that I'll show you a little bit more information about. Remdesivir is actually a molecule that was developed by Gilead and colleagues, to target the replication of Ebola virus. And it turns out that remdesivir has activity against SARS and MERS in animal models, and therefore it was tested for its ability to block SARS-CoV-2 infection in cell culture. And then based upon promising data in cell culture and some animal data, the molecule has been used in clinical trials that seem to be showing some efficacy of remdesivir in reducing the severity of disease in the most severely ill patients. And I think this is particularly promising because if one gives remdesivir earlier in the infectious process, then one might have a larger effect. Of course, there are some challenges with remdesivir. There are some liver toxicity issues that have been reported in the clinical trials that have been performed today. So one of the things that has been phenomenal about the global efforts in combating SARS-2 infection has been the pace of research and the speed with which discoveries have been made. Within 3 weeks of the sequence of the spike protein being published, Jason McClellan's group at the University of Austin in Texas solved by electron cryo microscopy the structure of the ectodomain of the spike protein. This is the protein that essentially catalyzes membrane fusion. It's a trimer. This is the piece of the protein that binds to the receptor. It's termed the receptor binding domain. This binds to ACE2. And of course, there are 3 copies of the receptor binding domain in the trimer. But in fact, that receptor binding domain can occupy 2 different conformations. A down conformation, which is not the ACE2 binding conformation, and an up conformation, which is the ACE2 binding conformation. And so this portion of the protein samples 2 different states. So it's quite a clever strategy in many ways for the virus, because it's not always exposing the key aspects of the receptor binding domain, which probably help facilitate evasion by the immune system of directing many antibodies towards this domain of the protein. If we compare the structure of this protein with that of SARS, we can see that this again is the up conformation, this is the down conformation. But if we look at the receptor binding domain compared to SARS-CoV, you can see that there's a lot of similarity, but there are some differences between the 2 protein structures. And they both engage ACE2 as the receptor, although SARS-CoV-2 binds the receptor ACE2 much more tightly. Another structural advance that has come is summarized in this study here by Yangao and colleagues, and that was published in Science a few weeks ago. And they had solved the atomic structure of the RNA-dependent RNA polymerase of SARS-CoV-2. There's nothing extraordinary about the RNA-dependent RNA polymerase. It looks essentially like the RNA-dependent RNA polymerases of all RNA viruses. The first one was solved in 1997, that of poliovirus. So we've known what these molecules look like for 20-some years. But the advantages of having this structure now is that it can really help inform drug development and advancing drug development. And in fact, in this study, what they show here in this is a model of remdesivir in the active site of the polymerase. And what the mechanism by which remdesivir functions is that it gets incorporated and then leads to a chain termination event by the polymerase as it's making RNA. And so if you, so remdesivir is presumably being incorporated by the SARS-CoV polymerase, and then precisely where chain termination occurs is not fully defined for SARS-CoV-2. In the case of Ebola, what was noted is that chain termination occurs a few nucleotides downstream, so that you can actually still add nucleotides on after the incorporation of remdesivir. And we don't yet know that for SARS-CoV-2. So there's a lot of work happening here on campus in basic science as well as clinical science. There are more than 35 groups who are studying aspects of the replication of this virus, trying to develop countermeasures and vaccines. Those groups are distributed largely among these departments, although I'm sure I've missed some departmental affiliations of some investigators working on this virus, so I apologize if anybody is left off. It wasn't intentional. But those investigators are studying virology, they're studying the pathogenesis of the disease, They're studying animal models of disease. They're trying to develop countermeasures, which include antibodies, small molecule inhibitors, and vaccines. And there's a lot of effort focused on developing nucleic acid and serological-based diagnostic assays. We need, as a country, we need millions of tests a week to help combat this infection, or at least that's one of the messages that's being put out by NIH these days. And so there's a lot of efforts on campus to really circumvent some of the supply chain issues that are out there with regard a lot of the existing commercial assays that report on viral presence by detection of nucleic acid from nasopharyngeal swabs, for example. There's a lot of effort on campus to try and advance saliva-based rapid diagnostic assays for the presence of virus, as well as a terrific effort on new serological assays. So I want to share a little bit about the work that's happened in my own group and some of the collaborative studies that we've been engaged in combating or trying to combat this virus. And I first want to step back and share a little bit about how we were working with Ebola virus a number of years ago. And the approach that we took was to take the genome of vesicular stomatitis virus, which is a livestock pathogen that is relatively innocuous for humans. This virus is endemic in Central America. In fact, it's estimated that about 30% of the population of Panama have neutralizing antibodies against this virus, but there's no evidence of significant human disease associated with this infection. The virus has been incredibly well studied over many years. It replicates extremely robustly. Within 8 hours, one infectious particle can yield 10,000 infectious particles from a single cell. The virus encodes 5 proteins. This is the surface protein, the glycoprotein that mediates attachment and membrane fusion. So in the 1990s, Heinz Feldman in Winnipeg, Manitoba, at the Canadian Public Health Agency BSL-4 lab, replaced the glycoprotein of VSV with the glycoprotein of Ebola virus. And he studied that virus under BSL-4 conditions and demonstrated, in fact, that that virus was attenuated compared to Ebola virus and was actually, when given to experimental animals, able to protect them against a lethal challenge with Ebola. That virus is, in fact, now a licensed human vaccine that's marketed and distributed by Merck. This approach of switching the glycoprotein in the genome of VSV is one that then we adapted in my own group, and we placed the Ebola glycoprotein also into the genome of VSV, but to study how Ebola mediates infection of cells. And if we look at these electron micrographs, this is how VSV enters cells. It binds to the surface of the cell and then gets endocytosed through a clathrin-coated pit. But if we now look at what happens to VSV Ebola virus, it enters through a very different mechanism. This is a macropinocytic uptake mechanism. And so we use this virus to actually probe how Ebola is entering cells. And the way that we did this is together with Tyne Brummelkamp, who was a fellow at the Whitehead Institute in Cambridge, Massachusetts. We did a haploid genetic screen. This predates CRISPR screens. And we took a haploid human cell line and performed insertional mutagenesis in this haploid human cell line on a random basis. And the mutagenesis was sufficiently, there was such an extent of mutagenesis that we could get insertions in about 98% of all expressed genes. What this then allowed us to do was to infect cells with VSV Ebola virus and select those cells that resisted infection. And then by sequencing the point of insertion, we could find out which genes had been inactivated through this mutagenesis. And so the cells, the hypothesis was that the cells that would survive infection would do so in a way that reflected the Ebola virus entry pathway. And in fact, that's exactly what happened. And we identified several host genes. And there are just two that I'd like to bring your attention to. One is the cholesterol transporter, Niemann-Pick-C1, which turned out to be an intracellular membrane protein that was required for viral infection. It was the receptor. And the other is this molecule here, PIC5, which is a kinase that I'll tell you a little bit more about. So Niemann-Pick-C1 is an endosomal cholesterol transporter. It's present in the late endosomes of all cells. It exports LDL-derived cholesterol from endosomes into cells. And in fact, there are human mutations in NPC1 that are associated with rare neurodegenerative disorders. We validated through a series of extensive experiments the role of NPC1 in infection. We showed that by inactivating NPC1, that the only virus that we protected cells from this panel of viruses, the only virus we protected against infection was VSV Ebola. Inactivation of NPC1 did not affect VSV rabies, VSV Borna disease virus, or VSV. Those viruses were still able to infect and kill these cells. I'll skip this slide in the interest of time. But we then went on to demonstrate in an in vivo model of Ebola pathogenesis, a mouse model, that mice lacking a single copy of NPC1 showed some resistance to a lethal challenge with Ebola virus. And this is just when they're missing one of the two copies. And they were completely resistant to a lethal challenge with Marburg virus. We published this work a number of years ago now. This is dating back to 2011. And at the same time as we published this study, a simultaneous publication from our colleague Jim Cunningham at the Brigham and Women's Hospital had used a chemical genetics approach that also converged upon Niemann-Pick-C1 as essential for Ebola virus infection. So I mentioned this molecule here, PIC5, that I'd just like to talk about a little bit further. So PIC5 converts PI3 phosphate into PI3-5P2. This is one of the least abundant phosphoinositides in the cell, and it plays a critical role in endomembrane homeostasis. So the reason that I'm talking about PIC5 is that there's a small molecule called apilamode, which has been tested as a potential therapeutic for Crohn's disease and has been given to patients over a prolonged period of time with a very good safety record. It's not an effective therapeutic for Crohn's, but it does mean that there is significant safety data regarding this molecule. So if you treat cells with apilamode and then infect them with VSV Ebola, you can see, based on this flow cytometry data, that the inhibitor, apilamode, suppresses infection. These are infected cells, shown by this rightward shift here on this flow cytometry plot. But in the presence of apilamode, we completely shift into the uninfected population. And we know that these are uninfected because we use another inhibitor, baflamycin, that we know will completely block Ebola infection. We see that these curves are superimposable. So we wanted to understand further how apilamode was blocking infection. And so we took this structural protein of VSV, the matrix protein, and fused it to GFP so that we could visualize the particles on their way into cells. I'll skip this slide in the interest of time. But if we block protein synthesis in infected cells, and what we're looking at now is an image of a single cell, if we block protein synthesis in infected cells and allow this virus to infect them, we can see that the incoming structural GFP protein migrates to the nucleus and lights up the nuclear membrane. And so this is a visual assay for successful entry and release of the contents of the virus into the cell. So we use that assay, and if you just focus on this bottom part of the slide, this is VSV Ebola virus. In the presence of this protein synthesis inhibitor, cyclohexamide, we see that the matrix protein gets released and goes to the nuclear membrane. If we use an inhibitor of PI3 kinase that suppresses partially the infection, we see that there's less successful infection. Many of these cells we don't see release. We see some release of the contents in some cells. Now, if we use baflamycin, this is the inhibitor of the vacuolar ATPase, we do not see efficient release. But if we use apilamode, we don't see release either. To look at this by live cell imaging, on the left here is a cell where we're seeing the release of these green dots are individual viral particles coming into a cell. And the matrix protein gets released and lights up the nuclear membrane. On the right-hand side is a cell treated with apilamode, and the particles stay stuck in these endocytic vesicles. We do not see release of the contents of the particles into the cell. If we also look at the location of these particles in relation to the viral receptor, NPC1, we can see again, in the absence of apilamode, we get successful infection, but in the presence of apilamode, the virus particles are stuck in the compartment with the receptor, but they're unable to release their contents into the cell. So this just shows in a little bit more detail the location of the particles in relation to markers of the endocytic pathway. This is early endosome antigen 1. And in the presence of apilamode, we can see association of the particles with early endosomal antigen 1 and NPC1. And we can see a shift further into the endocytic pathway over time. But those particles fail to release their contents in the presence of this molecule. So the reason that I did this detour into Ebola virus is we wanted to then examine whether or not apilamode would be successful in inhibiting SARS infection, SARS-CoV-2 infection. So we generated a VSV recombinant in which we replaced the spike protein of VSV with the spike protein of SARS-CoV-2 and rescued virus particles. When we initially did this experiment, we got a virus that grew extremely poorly. And therefore, we then selected, through forward genetics, a variant that grew much better so that we could study the virus readily in cell culture under BSL-2 conditions. What had happened in this process is that the virus had made a mutation that truncated the cytoplasmic tail of the SARS spike, which presumably facilitates more efficient incorporation into VSV particles. This, in fact, excuse me, this virus grows extremely robustly in cell culture. It grows to titers of up to 10 to the 8 infectious units per mil. And we can study the incorporation of the spike protein into the virus. This is VSV itself. These are the viral proteins, the polymerase protein, the glycoprotein. And you can see that in the VSV SARS-2 virus, the VSV glycoprotein is gone, and now instead we have the S1 and S2 proteins of SARS-CoV-2. This is an electron micrograph showing the incorporation of this spike protein into the VSV particles. So we use this as a surrogate to test the effect of a pill mode on infection. And what's shown here is that we efficiently suppress infection by VSV Ebola and VSV SARS-CoV-2 with 0.1 micromolar concentrations of apilamode. And this 1 micromolar suppresses infection to the threshold of detection in this specific assay. Together with Mike Diamond's group, we assessed the effect of apilamode on a clinical isolate of SARS-CoV-2. And this correlates, the inhibition of the clinical isolate correlates very well with what we've seen for the VSL2 VSV SARS-CoV. This work has been submitted. It's a collaboration between my group, Mike's group, and the group of Tom Kirkhausen. This manuscript was submitted and is presently under review. We've also, together with the Diamond Lab, used this reporter to compare the ability of monoclonal antibodies that have been generated, including some by RNAdependent group, and some by other investigators, to compare the ability of these monoclonal antibodies to neutralize the clinical isolate or the recombinant, the BSL2 reporter. And what's shown here is that there's quite a good correlation in the ability of these antibodies to neutralize both viruses. Similarly, using recombinant soluble ACE2 protein, the receptor, the human version of the receptor, inhibits, and this was produced by David Fremont's group, inhibits the clinical isolate. The mouse version of ACE2 does not inhibit, and we understand the basis for this distinction. And the same result is seen with the BSL2 VSV reporter virus. We've also compared the ability of these two viruses to be neutralized by a large panel of sera from patients who've recovered from infection. And what's shown here is both the clinical isolate and the BL2 reporter are neutralized by these sera. This is donor 1, donor 2, and donor 3, and you can see that these curves look remarkably similar. And if you do a correlative test over a large panel of sera, we can see that there's quite a good correlation. If we look at the outcomes, this is a collaboration between my group and a group at MGH. If we look at the outcomes of patients, we compared the sera from several COVID-19 positive patients and several negative patients, and all of the positive patients have neutralizing antibodies. And in fact, those with the most severe disease had the highest titers, which presumably reflects antigen load in those patients. So as I mentioned, there's quite a good correlation between the VSV BL2 reporter and the clinical isolate. And so we're quite excited that this represents a readily accessible BL2 reagent to probe neutralizing antibodies and inhibitors and biology related to the entry pathway of SARS-CoV-2 that we hope will be useful for defining inhibitors. So where are we on vaccines? There are many, many different vaccine candidates that have been tested, but I chose to use some figures that I took from this recent publication in Immunity by Florian Kramer's group, because I think it summarizes quite nicely the strategies and approaches that people are using towards vaccine development. So there are several different categories of vaccines that are being developed. There are RNA-based vaccines, including the one from Moderna that I'm sure people have heard of. There are DNA-based vaccines that people are testing, recombinant protein vaccines, viral vectored vaccines. David Curiel, together with Mike Diamond here, is testing a chimp ad vectored vaccine. And my group, together with Mike, is testing a VSV vectored vaccine. There are also approaches underway to test inactivated vaccines and live attenuated vaccines. The time frame for vaccine development is a hot topic at the moment. And this sort of goal, 6 to 18 months, is something that one hears a lot about. But I think one should put this in perspective. We've been trying to develop a vaccine for HIV since 1980, and we still don't have one. People have been trying to develop a vaccine for respiratory syncytial virus since the early 60s, and we still don't have one. And so, I think this is a very optimistic time frame. I hope that we can accomplish something like this in this time frame. And people have advocated all sorts of strategies for compressing aspects of this time frame. But I think realistically, 2 years is incredibly optimistic. Typically, vaccines have taken a decade to bring to market or more. So these are some of the platforms that are being evaluated. RNA-based vaccines. There are some advantages, no infectious virus. There are some disadvantages. One that's not listed here is that there are no licensed human RNA vaccines yet. DNA vaccines, there are several advantages. Again, there are some disadvantages. Recombinant protein vaccines. So there are efforts underway to use recombinant S protein. There are advantages. No infectious virus is needed. You can use adjuvants to increase immunogenicity, but this is a laborious way to generate a vaccine, and there are challenges to produce full-length recombinant ectodomain of the protein in large quantities, and that has been a problem for this approach. There are viral vector-based approaches. There is a VSV vectored Ebola vaccine that has been licensed and is produced by Merck. There are advantages to this. You don't need to handle the wild type virus, and you can just use the S protein of the SARS-CoV-2 and express it in a different viral vector. So there are adenovirus vectors that are being tested by a number of groups too. There are some disadvantages. Pre-existing immunity to the vector might negatively affect the vaccine's effectiveness. Live attenuated vaccines. This approach has been used effectively for a number of viruses. Those include measles and polio. The need for the extensive safety testing is always needed for a live attenuated vaccine. And in fact, the polio vaccine that was very successfully developed by Jonas Salk is an inactivated vaccine, but there was a second vaccine that was developed by Albert Sabin, which is a live attenuated vaccine. And both of those vaccines have been very effective in eliminating or almost eliminating polio from the globe. There are advantages and disadvantages to each. The live attenuated vaccine, the disadvantage is that safety testing and reversion of the live attenuated vaccine can be a problem. So we're hoping that with the, so as I mentioned, Merck has used the VSV Ebola vaccine, developed the VSV Ebola vaccine that is licensed for use in humans. And we're hoping that the VSV recombinant that I mentioned that we've generated that has the SARS-CoV-2 spike on the virus will also be a potential vaccine candidate. And together with Mike Diamond, we have put this virus into mice. And in fact, we should get the first data on the immunogenicity of this virus in mice today. So I'd just like to acknowledge a very large number of people that are shown here who've been involved in some of the aspects of the work that I have talked about today. And I'd also like to acknowledge funding sources. And with that, I'll stop. And if there are any questions, I'd be happy to answer them.
Are you able to hear me?
Yes.
Okay.
Hi.
Sorry, technical difficulties, but I'll try one more time. So the question that was proposed from the group is, what is the evidence of tocilizumab and other IL-6 inhibitors as possible interventions?
So those inhibitors are designed to try and counteract the immune response and the immune pathology of disease. I'm not so familiar with the current state of where those inhibitors are, but certainly there are a number of trials that are ongoing with them. But I can't comment further because I'm not as up to date with what's happening with the IL-6 inhibitors. But they don't target the virus, they're targeting the host response and therefore trying to reduce the pathology.
Okay. Another question. That was a terrific talk, thank you. What are the biggest challenges for the VSV versus adenovirus vaccine models?
So I think every vaccine candidate has really significant challenges. There are significant hurdles for every single one of them. And that's why it's great news that there are 100 and something different vaccine candidates that are being tested. Because we're going to need a lot of different vaccines. One of the big challenges is going to be safety. So I'm sure that VSV and I'm sure adenovirus will both be immunogenic and elicit the production of neutralizing antibodies. Hopefully, the production of neutralizing antibodies is sufficient to protect against disease, and that there isn't any evidence of antibody-dependent enhancement of disease. We don't think that there will be, but we do not know this definitively yet. I think, you know, Safety issues are going to be an important thing, demonstrating that these vaccines are safe. The rotavirus vaccine, when it was first released, was withdrawn after the vaccine was given to 1 million recipients. And so I think, safety testing of vaccines is always a key challenge. And I think that's going to be one of the issues that we will deal with. It's hoped that some of the strategies, like using RNA-based vaccines, that there will be less safety issues. But at the moment, we just don't know this, and we don't know, there are no licensed human nucleic acid-based vaccines. So I think we need lots and lots of different candidates.
And then in the interest of time, I'm going to pick one or two more questions for you. One of them comes about the camostat mechanism, and the question being, is it good enough to be useful?
So camostat's an interesting candidate. The reason, will camostat alone work? I'm not sure that it will work alone, because if you block the protease at the cell surface, TMPRSS2, maybe the virus can just then enter cells via endocytosis, and the intracellular proteases will cleave the spike and allow membrane fusion to occur. And so maybe the right strategy is to combine something like apilamode and camostat as a potential combination therapy. I don't know that that's happening. I know that there are plans to do a trial of apilamode with remdesivir, but I don't know that there is a planned approach yet with camostat. But I think that could be quite an interesting approach.
Okay, last question just from time's sake. So can you speculate as to whether protease inhibitors might be effective when administered early in the course of infection? The published RCT appears to only include patients treated during late time points around 12 to 14 days after infection.
I think administration of, remdesivir has worked late in infection, but I think it's a very good point. There are many inhibitors that one would want to give much earlier in the course of infection, where one is interfering with virus replication before the virus has already taken hold. And I think that even though studies with protease inhibitors have not actually had much significant clinical benefit, that looking earlier in the course of disease is probably the right strategy, where one has more chance to interfere with actual virus replication.
All right, Dr. Whelan, thank you so much for taking time today to talk to us. We really appreciate you coming and discussing some of the research behind coronavirus here. We have another session next week, so stay tuned for next week, where we look at some of the clotting associations with COVID-19. So we'll see you all then. And again, Dr. Whelan, thank you so much for joining us today.
Thanks, it was my pleasure. Thank you.